Good evening, everyone. So, we'll start in 5 minutes so that more people can join in. So, most likely, what we are going to do today is, as usual, it's been a while since we had a revision or recap sort of a thing. So, we might quickly go through the revision and see whatever the things we have covered. And then we're gonna continue our discussion on complement system. And if the time permits, then we'll have little bit of questions which will be pertaining to complement system and the current week things which were covered in the lecture videos. Okay, so let's wait for 5 minutes so that more people can join in and then we can start. Okay. I guess we can start our session now. So, yeah, this might seem a little bit primitive and repetitive, but the goodness of revision lies in repetition itself. Okay, so we're gonna revise very quickly so that you guys get the brush up of the things which you have already covered, as well as if you had any doubts or any questions in making any connections between the separate topics, that might also be resolved in the meanwhile. Okay, so we started like the subject itself, then the definition of it. So the term immunity usually refers to protection against infectious pathogens. However, reactions to some non-infectious substances, including harmless environmental molecules, tumors, and even one's own molecules, are also considered forms of immunity. The collection of cells, tissues, and molecules that mediate these reactions is called the immune system, and the coordinated response of these cells and molecules to pathogens and other substances make up an immune response. So, the importance of immune system in health lies in some of the roles of immune system. So, defense against infections, defense against tumors, control of tissue regeneration and scarring, cell injury and pathologic inflammation, recognition of and injury of tissue grafts and newly introduced proteins. Infection and immunity. The most important physiologic function of the immune system is to prevent and eradicate infections. Individuals with defective immune responses are at increased risk of serious, often life threatening infections. Conversely, stimulating immune responses against microbes through vaccination is the most effective method for protecting individuals against infections. This approach has led to the worldwide eradication of smallpox, an only disease that has been eliminated from civilization by human intervention. And you can see in the table how well the vaccine works. Then the stages of host defense, innate and adaptive immunity. So the defense against infections is provided by the early reactions of innate immunity and the later more powerful reactions of adaptive immunity. Innate immunity, also known as natural and native immunity, is present in all healthy individuals at all times. First line of defense blocks microbial entry and eliminates invaders rapidly, provides immediate response upon infection, does not require prior exposure to pathogens, evolutionarily older defense mechanism. When we talk about adaptive immunity, it is activated after exposure to microbes, requires proliferation and differentiation of lymphocytes, highly specific to the invading pathogen, involves immunologic memory, stronger responses on re-exposure. Takes a few days to develop and more specialized and potent due to clonal expansion and effector diversity. This is the overall components of innate and adaptive immunity, in which in innate immunity you have mast cells, phagocytes, dendritic cells, complement system, and NK cells and ILCs. While in adaptive immunity you have B lymphocytes and T lymphocytes, which at the end, or say after maturation and proliferation, makes plasma cells and effector T cells. Then we discussed about the components of innate immunity. So it is the first line of defense: epithelial barriers, skin and mucosal tissues, antimicrobial substances from epithelial cells, resident immune cells in or under epithelia. The basic function lies in blocking the entry of the microbe. The second line of defense, that is formed by the phagocytes, that is neutrophils and macrophages, plasma proteins including the complement system, and the function lies in destroying the microbes in the tissue which has just entered, or especially which are present in the circulation. And the additional roles involve, it triggers or informs the adaptive immune response or adaptive immune system, recognizes limited shared microbial structures, pathogen associated molecular patterns, that is PAMPs. The adaptive immune response is mediated by lymphocytes with highly diverse and variable receptors for foreign substances and the products of these cells, such as antibodies and other proteins. Adaptive immunity, some key features include: essential for defense against pathogens that evade innate immunity. Lymphocytes express specific receptor to detect microbial and non-microbial molecules. Recognizes a wide range of antigens. What are antigens? Antigens are any molecule specifically recognized by lymphocytes or antibodies. Then we looked into the types of adaptive immunity, where we discussed about that there are basically two types of adaptive immunity, called humoral immunity and cell-mediated immunity, mediated by different cells and molecules that provide defense against microbes in different locations. Extracellular microbes, which survive outside host cells and are readily destroyed when they are ingested by phagocytes, are combated by antibodies. Microbes that have evolved to survive inside host cells, either in phagocytic vesicles or in the cytosol, are eradicated by the actions of T lymphocytes. So the humoral immunity and the cell-mediated immunity. Cell-mediated immunity can have two pathways, one via CD8 positive T cells and one via CD4 positive T cells. And in humoral immunity, majorly it will be the antibody things, or triggered by the B lymphocytes. So humoral immunity is mediated by proteins called antibodies, which are produced by cells called B lymphocytes. Secreted antibodies enter the circulation, extracellular tissue fluids, and lumen of mucosal organs such as the gastrointestinal and respiratory tracts. Antibodies defend against microbes present in these locations by preventing them from infecting tissue cells and by neutralizing toxins made by the microbes. Antibodies also enhance the uptake of extracellular microbes into phagocytes, resulting in the killing of the pathogens. In addition, antibodies are transported through the placenta into the fetal circulation and protect the fetus and newborn from infections. If we talk about cell mediated immunity or CMI, defense against microbes that have entered host cells, mediated by T lymphocytes, critical for intracellular pathogens. Limitations include prevent microbial entry but also ineffective against microbes who has already entered the host cells. Then different types of T lymphocytes in cell mediated immunity includes helper T cells. They basically produce cytokines, activates phagocytes to destroy ingested microbes in intracellular vesicles, also recruit phagocytes to infection sites to combat extracellular microbes. Cytotoxic T lymphocytes directly kill infected host cells, including non-phagocytic cells, especially effective against viruses and cytoplasmic pathogens. Immunity may be induced in an individual by infection or vaccination, or conferred on an individual by transfer of antibodies from an actively immunized individual. When we talk about types of adaptive immunity, so the adaptive immunity means developed after the exposure to a microbe or its antigens. Individual mounts an immune response and eliminates the infection, provides long lasting resistance. Exposed individual becomes immune, unexposed individuals are still considered naive. While the passive immunity, antibodies are transferred from an immune individual or produced in labs, provides immediate but short term protection, does not include immunologic memory because the infection has not taken place in the host. Physiological examples include newborns who receive maternal antibodies via placenta or via the breast milk. Clinical applications can be summarized as in immunodeficiency disorders. In these, the patients are generally treated with pooled antibodies. Emergency treatment for infections such as SARS-CoV-2 and snake bites. Monoclonal antibodies used in cancer immunotherapy. T cell transfer is not feasible between unrelated individuals. Properties of adaptive immune responses. Features: specificity, diversity, memory, and non-reactivity to self. When we talked about clonal selection, these basically means that which type of antibodies will be selected. So there are 4 basic steps which are required in the clonal selection. So in this, the lymphocyte clones with diverse receptors arise in generative lymphoid organs. Clones of mature lymphocytes specific to many antigens enter lymphoid tissues. Antigen specific clones are activated or selected by the antigens, and then antigen specific immune responses occur as a result of this clonal selection. Other features of adaptive immunity includes adaptive immune responses have other characteristics that are important for their functions. Immune responses are specialized and different responses are designed to defend best against different kinds of microbes and at different sites of infections. For example, there are several classes of secreted antibodies, each of which perform a different set of functions, and there are several subsets of T cells, each of which combats infections in different ways. All immune responses are self-limited and decline as the infection is eliminated, allowing the system to return to a resting state, prepared to respond to another infection. Cells of the adaptive immune system. So lymphocytes, they are basically 2 types of lymphocytes, B lymphocytes and T lymphocytes. So B lymphocytes, they are the mediators of humoral immunity, while T lymphocytes are the mediators of cell mediated immunity. Myeloid cells, they include neutrophils, macrophages, dendritic cells and mast cells. So neutrophils and monocytes or macrophages, they phagocytize the killing of microbe. Dendritic cell, antigen, they generally present antigen to T cells. Initiation of T cell responses. Mast cells, secretion of inflammatory mediators. When we talk about the primary and secondary immune responses, we learned that the primary response is generally has a lag phase associated with it. The strength is not as high as compared to the secondary response. In the primary response, majorly Ig, M type of antibodies are produced, while in secondary response IgG, and depending upon the infection, those antibodies are produced. Such type of infections responses completely depend upon the antigen. So it is not necessary that if secondary response is going for antigen X, it can't initiate the primary response for antigen Y. Okay. Then we talked about the classes of lymphocytes. So there are basically a few types of lymphocytes, as we have seen in the previous lecture, B lymphocyte and T lymphocyte. But even in T lymphocytes, there are different types of T cells. So the B lymphocytes, the major function of T lymphocyte is neutralization of microbe, phagocytosis, complement activation. While T helper cell or helper T lymphocyte, they how do they function basically is activation of macrophages, inflammation, and activation of B lymphocytes. Cytotoxic T lymphocytes, they kill the infected cell. Regulatory T lymphocytes, they basically lead to the suppression of the other lymphocytes after the infection has been cleared. When we talk about the features or specificity and receptors of innate immunity and adaptive immunity, we see that the specificity of receptors of innate immunity is not that high in comparison to the adaptive immunity. The receptors for innate immunity, they are majorly located in plasma membrane, endosomal membrane, or cytosol. While for adaptive immunity, the receptors are located majorly and unequivocally on the plasma membrane. When we talk about the numbers and types of receptors, there are 100 different types of receptors in innate immunity, while there are only two major different types of receptors in adaptive immunity. One is immunoglobulin, which is a B cell receptor, and T cell receptor. Okay. Then we had a brief discussion about the acute inflammatory responses, which is basically the like a circadian kind of a thing. It's a chain like a reaction that one thing leads to the other. So in this, the cytokines and other mediators are produced by macrophages, dendritic cells, mast cells, and other cells in tissue in response to microbial products and damaged host cells. Some of these mediators, example, histamine, prostaglandins, increase the permeability of blood vessels, leading to the entry of plasma proteins. And in plasma protein, we have complement proteins, okay, into the tissues. And others, such as interleukin 1 or TNF, they increases the expression of endothelial adhesion molecules and chemokines that promote the movement of leukocytes from the blood into the tissue, where the leukocytes destroy microbes, clear damaged cells, and promote more inflammation and repair. Then we saw the sequence of events in migration of blood leukocytes to the site of infection. And why this is happening? This is happening because of the inflammation. So at sites of infection, macrophages, dendritic cells, and other cells have encountered microbes, produce cytokines such as tumor necrosis factor and interleukin 1 that activate the endothelial cells of nearby venules to express selectins and ligands for integrins and to secrete chemokines. Selectins mediate weak tethering and rolling of blood neutrophils on the endothelium. Integrins mediate firm adhesion of neutrophils, and chemokines activate the neutrophils and stimulate their migration through the endothelium to the site of infection. Blood monocytes and activated T lymphocytes use the same mechanism to migrate to the site of infection. Then we discussed about the cellular locations of receptors of the innate immune responses. So some receptors such as certain toll-like receptors or TLRs and lectins are located on the cell surface. Other TLRs are in endosomes. Some receptors for viral nucleic acids, bacterial peptides, and products of damaged cells are in the cytoplasm. NOD and RIG refers to the founding members of families of structurally homologous cytosolic receptors for bacterial and viral products respectively. There are 5 major families of cellular receptors in innate immunity. First is TLRs, that is toll-like receptors. then C-type lectin receptors, then NOD-like receptors, then RIG-like receptors, and cytosolic DNA sensors. Now, how does the activation and the function of macrophage occurs? In innate immune responses, macrophages are activated by microbial products binding to TLRs and by cytokines such as NK cells derived interferons gamma, which lead to the production of proteins that mediate inflammatory and microbial functions of these cells. Cell surface complement receptors promote the phagocytosis of complement coated microbes as well as activation of the macrophages. Macrophage FC receptor for IgG binds antibody coated microbes and perform functions similar to those of the complement receptors. Then we had a brief discussion about antigen recognition in the adaptive immune system, basic structure of lymphocyte antigen receptors and development of immune repertoires. So B and T lymphocytes express different receptors that recognizes antigen. Membrane bound antibodies on B cells and T cell receptors on T lymphocytes. Some questions needs to be answered. So how do the antigen receptors of lymphocyte recognize extremely diverse antigens and transmit activating signals to the cells? What are the differences in the recognition properties of antigen receptors on B cells and T cells? How is the vast diversity of receptors in the lymphocyte repertoire granted? The diversity of antigen recognition implies the existence of many structurally different antigen receptor proteins. More than can be encoded in the inherited genome. Therefore, special mechanisms must exist for generating this diversity. Then we looked into the antigen receptors of lymphocytes, especially the properties of antibodies and T cell antigen receptors. So antibodies, also called as immunoglobulins, may be expressed as membrane receptors or secreted proteins. TCRs only function as membrane receptors. When immunoglobulin or TCR molecules recognize antigens, signals are delivered to the lymphocytes by proteins associated with the antigen receptors. The antigen receptors are associated signaling proteins from the B cell receptors and TCR complexes. The single antigen receptor are shown recognizing antigens, but signaling by BCRs typically requires the binding of two or more receptors to adjacent antigen molecules. Now we know what we are talking about here, right? The co-receptors concept. But signaling by BCR typically requires the binding of two or more receptors to adjacent antigen molecules. The important characteristics of these antigen recognizing molecules are summarized. Some of these are discussed, like later we have discussed. When we talk about B cell receptors, so they are membrane bound antibodies, recognize diverse chemical structures, proteins, lipids, carbohydrates and nucleic acid, and small molecules as well. Detect native shapes, conformations of macromolecules. Activated B cells secrete antibodies with identical specificity. Function: bind, eliminate microbe and toxins in humoral immunity. T cell receptor, it recognizes only peptides bond to MHC molecules. Antigens are from intracellular proteins to extracellular proteins processed by APCs. Do not bind free or soluble antigens. Functions are target infected tumor or antigen presenting cells. Key differences between B cell and T cells lies in the way they are recognizing the antigen. Structure of antigen receptor molecules. So the domain organization, the variable region, that is the antigen recognition, differs between lymphocyte clones. Constant region, there is structural integrity and binds to signaling molecules conserved across clones. Variation is concentrated in the hypervariable region or the CDRs. Functional design, so basically their variations are limited to small regions. And the genetic basis, that special mechanisms in developing lymphocyte generate diverse V regions for unique antigen receptors. association with invariant protein. So antigen receptors polypeptide pair with invariant membrane proteins, deliver intracellular signals after antigen recognition. Variability in receptor signals in receptors, signal transduction via conserved proteins. Then B cell receptors and antibodies so exist in two forms, membrane bound and secreted antibody. You know, they are commonly called Igs or immunoglobulins. They basically recognize as antigens or toxins in their native form, while T cell receptor, they only exist as membrane receptor and they are not secretory in nature, and the effector phase and cell mediated immunity performed by T cells and other leukocytes that are activated. Okay, now we had a discussion about antibodies. So an antibody molecule is composed of 4 peptide chains and 2 identical heavy chains and 2 identical light chains, with each chain containing a variable region and a constant region. So I'm gonna share this video again, so because it's very important to be very clear about these topics. Yeah, so this is all the revision, these are all the things which we have already studied about. It's just I'm helping you revise so that in exams you don't have to put a lot of effort in like going back and forth.
Discuss B cell receptor. B cell antigen receptors are immunoglobulins expressed on the surface of B cells. The receptor is produced terminally differentiated by B cells or plasma B cell and secreted as antibodies into the blood. This is extracellular region of B cell. This is cell membrane. While this is cytoplasmic region of B cell. Immunoglobulins are the glycoproteins, composed of 2 identical heavy and light chain. The weight of heavy chain is 50,000 to 70,000 Dalton, while the weight of light chain is 25,000 Dalton. There are 2 types of light chains denoted by kappa and lambda. Cysteine residues form bridge between individual chains of immunoglobulin molecule. This is antigen binding fragment or Fab, while it is crystallizable fragment or FC fragment. They are separated by enzyme called as papain. FC fragment possessed binding sites for complement factor C1q. Light chains consists of 2 regions approximately of equal size. This is variable region of light chain, subdenoted by VL. And this is variable region of heavy chain, so it is denoted by VH. This is constant region of light chain, so it is denoted by CL. And constant region of heavy chain, so denoted as CH, which is further divided, so denoted as CH1, CH2, and CH3. The constant region varies little from one immunoglobulin to another immunoglobulin. There is huge variability among amino acid sequence in variable region. Both constant and variable region consists of about 110 amino acids. Look at the heavy chain. It has 1 variable region with 110 amino acid and 3 constant regions. But in case of immunoglobulin M and immunoglobulin E, there are 4 constant domains. Don't worry about the structures of immunoglobulins. They are also explained in detail later in this video. But remember that immunoglobulin M and immunoglobulin E have 4 constant regions. Due to the variability among amino acids, in variable region, immunoglobulin variable portion varies from one immunoglobulin to another immunoglobulin and involve an antigen binding. This is N-terminal. This is C-terminal. Now, C-terminal of B cell receptor has spacer region having 26 amino acids. Then, this is transmembrane portion having 25 amino acids. Then there is short cytoplasmic region, which is very short, having 3 amino acids. Cytoplasmic region of B cell is so small that it cannot activate B cell or cannot give signal to B cell for activation. So, for proper activation of B cell, there are some other components, which is A, heterodimer. Heterodimer means having 2 different monomer. It has one monomer called as Ig alpha, and other is Ig beta. The cytoplasmic portion of Ig alpha and Ig beta have ITAM or immune receptor tyrosine activation motif. It has amino acids called as tyrosine. This region has kinase enzyme. So this tyrosine is phosphorylated or phosphate is added to tyrosine through kinase enzyme. Now, signaling starts and leads to B cell activation. This was all about today's session.
Correct? So this is the structure of antibodies. As you can see, here it is the IgG antibody, here it is the IgM antibody. This is the real crystal structure of the antibody, and this is the representation of the domains which are making the this part of the antibody. Okay, then some of the features of the major isotypes of antibodies include Ig, like are IgA, IgT, IgE, IgG and IgM. So IgA majorly functions in the mucosal immunity as a neutralizing agent. IgD, they are like for naive B cell antigen receptors. IgE, they are immediate hypersensitivity and mast cell activation. IgG are involved in defense against microbes in all tissues, neutralization, opsonization and phagocytosis, complement activation, antibody dependent cellular cytotoxicity and neonatal immunity as well. IgM, naive B cell antigen receptors, monomeric form, defense against microbes, includes complement activation and neutralization. So we will see some of the antibodies function and antibodies as whole.
Of antibodies or immunoglobulins, named as immunoglobulin G or IgG, immunoglobulin M or IgM, immunoglobulin A or IgA, immunoglobulin D or IgD, and immunoglobulin E or IgE. All these classes have the basic 4-chain antibody structure. Also, they all have 2 types of light chains named as kappa and lambda. Both types are very similar in structure and no functional difference as well, but are coded for by different genes. Difference between these classes comes in heavy chain that are different for each class of immunoglobulins. They are termed as gamma for IgG, mu for IgM, alpha for IgA, delta for IgD, and epsilon for IgE. The differences among antibody classes are most pronounced in the Fc regions of the antibody classes, and this leads to the triggering of different effector functions on binding to antigen. For example, IgM recognition of antigen might lead to complement activation, whereas Ig recognition, possibly of the same antigen, might lead to mast cell degranulation and anaphylaxis. Structural differences are also very pronounced among different classes of antibodies. The major antibody in the serum is IgG, and as this is the best understood antibody in terms of structure and function, so we shall consider it first. The other antibody classes will be considered in relation to IgG. So let's look at different classes of antibodies one by one. Starting with immunoglobulin G. This is the most abundant antibody in serum. as it constitutes approximately 75% of total serum immunoglobulins. It is the simplest of all antibodies in terms of structure. It consists of similar 2 light chains and 2 heavy chains. The light chains can be kappa or lambda, and the heavy chain present in I, GG, is represented as gamma. IgG has 4 subclasses in humans, that are IgG1, IgG2, IgG3, and IgG4, having gamma 1, 2, 3, and 4 respectively. The different IgG subclasses have slightly different structures and functions, although they are closely related to each other. Some important functions of IgG include: it is the only class of immunoglobulin capable of crossing the placenta. Consequently, it provides some degree of immune protection to the developing fetus. Among its subclasses, only IgG3 and IgG4 can cross placenta. The Fc portion of IgG can bind with complementary proteins. Mainly, it can bind avidly with C1q, an important protein that cause complement activation. Of the 4 subclasses, Ig1 and IgG3 activate complement most efficiently and are responsible for clearing most protein antigens. It can also bind to the phagocytic cells once they are attached with antigen, thus making engulfment of that invader easy by phagocytic cells, under a process called opsonization. IgG1 and IgG3 are also efficiently involved in opsonization, thus facilitates the process of phagocytosis. But IgG2 and IgG4 react predominantly with carbohydrate antigens and are relatively poor opsonin. IgG can also attach with Fc receptors present on monocytes, macrophages, and other immune cells and aids in their activity against foreign antigen. Next class of antibody is immunoglobulin M.
I work better under pressure. If you keep telling yourself this, but still end up rushing and stressed, then listen closely. The truth is, you don't work better under pressure. You just never felt safe to start early. Every time you sit down to begin, your brain distracts you, scrolling reels, reorganizing your desk, or randomly taking a nap. That is not laziness. It is your brain's coping mechanism, avoiding discomfort because it links with fear of failure and not being perfect. So you delay, you avoid, then you panic and rush at the last minute. But tell me, how long will you keep living like this? Hi, I'm Dr. Meghna Dikshit, a brain and performance expert. And for the last 28 years, I have been helping people rewire their minds and transform their lives. And now, I want to help you. Join my 3-hour Ultimate Success Builder workshop, where I will teach you the exact techniques to reprogram your brain and break the habits that are secretly holding you back. In this workshop, you will learn how to train your brain to work smarter, not harder. Overcome mental resistance and avoid procrastination. Rewire your subconscious to become productive and focused. It is high time you become the most productive version of yourself. So click the link below and book your seat.
IgM is the first antibody produced in response to a microbial attack by B cells. It circulates in the blood and lymph and constitutes 6% of the total antibody content in the serum. Structurally, it consists of a pentamer of 5 immunoglobulin molecules joined together by disulfide bonds, making it a largest antibody among all classes. Here, an extra protein chain called as joining or J chain is present that is attached to the heavy chain of 2 of the 5 immunoglobulin molecules that make up IgM. This J chain is required for the proper polymerization of IgM into pentamers, and in the absence of a J chain, pentamers are not formed. So this pentameric structure provides 10 antigen binding sites to one IgM molecule. Although steric hindrance is there, that practically allows it to use 6 antigen binding sites at once. These large number of antigenic sites facilitates efficient activation of immune system. IgM is extremely efficient at binding and agglutinating microorganisms early in inflammatory response. It also plays a role in opsonization, thus helping in the process of phagocytosis. It is also very efficient at making larger immune complexes, which can activate unwanted inflammation and ultimately an inflammatory disease. That's why its production is downregulated as soon as sufficient IgG has been generated. It has 5 complement binding sites that makes it efficient in complement-mediated lysis of the organisms. Next class of immunoglobulin is IgA. It is the major antibody found in body secretions like tears, sweat, saliva, breast milk, serum, contents of lungs, gut, urine, etc. It is also present in mucosal lining of respiratory and intestinal tract, making it a major mucosal immunoglobulin. It constitutes 13% of the total antibody content in the serum and is divided into 2 subclasses that are IgA1 and IgA2. It exists in both monomeric as well as dimeric forms. In dimeric form, it is composed of 2 H molecules, one joining chain attached with heavy chains of 2 immunoglobulin molecules, and a secretory piece. Attachment of secretory piece prevents digestion of this immunoglobulin in the intestinal and bronchial secretions. IgA2 is the predominant subclass in secretions and neutralizes antigens that enter via these mucosal routes. IgA1, the monomeric IgA in serum, is capable of neutralizing antigens that enter the circulation, but IgA1 is sensitive to bacterial proteases and therefore less useful for host defense at mucosal surfaces. IgA has additional functions via its Fc receptor, present on mononuclear cells and neutrophils, for activation of phagocytosis, inflammatory mediator release, and antibody-dependent cell-mediated cytotoxicity, ADCC.
मैंने ऐसी छोटी छोटी अंधे लड़कियां देखी.
Another class of immunoglobulin is IgD. It is present as a monomer and weighs around 180 kilodalton. It comprises less than 1% of the total antibody content in serum. IgD generally has a low serum concentration and is unstable in serum, being quickly degraded by serum plasmin. IgD is synthesized by antigen-sensitive B lymphocytes, is not secreted, acting as a cell surface receptor for activation of these cells by the specific antigen. It exists as a monomer of the basic Ig structure, but the functional significance of serum IgD is not clear. The last class of immunoglobulin is IgE. IgE is present in the least amounts, around 0.02% of the antibody content in the serum. These are present in the linings of the respiratory and intestinal tracts and respond to allergic reactions. Each molecule of IgE consists of one 4-chain unit and so has 2 antigen-binding sites, like the IgG molecule. However, each of its H chains has an extra constant domain, CH4, which confers on IgE the special property of binding to the surface of basophils and mast cells. When antigens bind to these attached IgE molecules, the cell is stimulated to release chemicals such as histamines that are involved in allergic reactions. Ig antibodies also help protect against parasitic infections. So this was all. After knowing
right, so now we're gonna go a little bit faster for our revision so that we can cover. So now we are going to be a little bit more faster so that we can cover the whole revision till complement system. Okay, then we had a word we discussed about the monoclonal antibodies. So monoclonal antibodies are like each B cell clone makes antibodies of one specificity. This principle is used to create monoclonal antibodies. So firstly, the how they are basically made. So this topic was not much discussed in the class, so this is just the overview of everything. Okay, B cells collected from an animal immunized with antigen. Short-lived B cells are fused with the immortal myeloma cells. These hybrid cells or hybridomas, they survive in selective medium due to the enzyme complementation. So hybridomas, they basically grow indefinitely, which is the part of this myeloma cell property, and they produce antibodies because they have this B cell property associated with them. The screening basically selects the hybridomas producing desired antibodies, which finally resulting in the monoclonal antibodies. So monoclonal antibodies are homogeneous, specific to a single epitope, useful in clinical medicine and research, obviously. So the challenges with mouse monoclonal antibodies are there because at the end of the day they are foreign for us. Okay, so they are produced by fusing mouse B cells with mouse myeloma cells. Human immune system recognizes mouse Ig as foreign, hence there will be an immune response against these monoclonal antibodies. They cannot be used for repeated injections in humans because otherwise our system will generate the memory of these cells and it won't let these cells be as effective as they should. So this problem was addressed by genetic engineering. So basically in this we make humanized antibodies, so the mouse antigen binding region plus human Ig framework, so they are less immunogenic and they are good for therapy, immunotherapy basically. So the clone DNA encoding human antibodies of desired specificity. Mouse immunoglobulin genes are replaced with human immunoglobulin genes, and the immunization yields specific human antibodies which are specific to humans. Okay, then this is the basic production of hybridomas and monoclonal antibody. So in this procedure, spleen cells from a mouse that has been immunized with a known antigen are fused with an enzyme deficient myeloma cell line that does not secrete its own immunoglobulins. The fused cells are then placed in a selection media that permits the survival of only immortalized hybrids. The normal B cells provide the enzyme that the myeloma lacks, and the unfused B cells cannot survive indefinitely. These hybrid cells are then grown as single cell clones and tested for the secretion of antibodies of the desired specificity. The clone producing this antibody is expanded and becomes a source of monoclonal antibody finally. So the basic steps, as you can see here, so we basically isolate the spleen cells from the mouse which is immunized with the antigen X. So here antigen X will be any antigen for which we want antibody against. Okay, then there will be a fusion of spleen cells and the immortal myeloma cells. Then only the few cells or the hybridomas, they will grow in this selection media. Then screening of these supernatants of each clone will be tested, and the hybridomas producing monoclonal antibodies will be selected. Then we had a brief discussion about the T cell receptors. So the TCRs, which recognizes peptide antigens displayed by MHC molecules in a membrane bound heterodimeric protein composed of A chain and a B chain, each chain containing one V region and one C region. The V and C regions are homologous to immunoglobulin V and C regions. In the V region of each TCR chain, there are three subvariables or variables or complementarity determining regions or CDRs at the end of the day. As in antibodies, CDR3 is the most variable among different TCRs. The C region of both A and B chain continue into short hinge region, which contains cysteine residues where a disulfide bond links the two chain, followed by the hydrophobic transmembrane domain. So this is the basic structure of TCR or T cell antigen receptor, and you can see with the help of MHC how it recognizes the processed antigen or the processed peptide. Okay, by the way, this is the real structure or the crystal structure of the antibody. This is how they look under the microscope, or say when we see them physically. Then we had a discussion about selection of major histocompatibility complexes or MHC, restricted CD4 positive and CD8 positive T lymphocytes. The strength of recognition of peptide MHC complexes by immature double positive T cells in the thymus determines the development of these cells into single positive CD4 positive and CD8 positive cells. So now you all have a huge and great idea about why we need these checkpoints. So we have checkpoints at B cell receptor, we have checkpoints at MHC receptors, and this is how our body is responding against to the infections. Okay, so there is both positive as well as negative selection goes into selecting what goes out, right? What is basically recognizing the antigen, how it is being processed, and all those things. So the summary of the adaptive immune system incorporated with that the molecules responsible for specific recognition of antigens are antibodies and T cell antigen receptors. Antibodies, also called Ig, may be produced as membrane receptors of B lymphocytes and as proteins secreted by antigen stimulated B cells that have differentiated into antibody secreting plasma cells. The T cell receptors or TCRs are membrane receptors and they are not in secretory in nature. The core structure of antibodies consists of two identical heavy chains and two identical light chains, forming a disulfide linked complex. Each chain consists of a variable region, which is the portion that recognizes antigen, and a constant region which provides structural stability, and in heavy chains perform the effector function of antibody. T cell receptors consist of an A chain and a B chain. Each chain contains one variable region and one constant region, and both chains participate in the recognition of antigens, which for most T cells are peptides displayed by the MHC molecules. The V region of immunoglobulin or Ig and TCR molecules contain hypervariable segments, also called complementarity determining regions or CDRs, which are the regions of contact with the antigen. The inherited genes that encode antigen receptors consist of multiple segments separated in the germline and brought together during maturation of lymphocytes. In B cells, the Ig gene segments undergo recombination as the cell mature in the bone marrow, and in T cells, the TCR gene segment undergo recombination during maturation in the thymus itself. So the receptors of different specificities are generated in part by different combinations of V, D and J gene segments. The process of recombination introduces additional variability in the nucleotide sequences at the site of recombination by adding or removing nucleotides from the junctions. The result of this introduced variability is the development of a diverse repertoire of lymphocytes, in which clones of cells with different antigen specificities express receptors that differ in sequence and recognition, and most of the differences are concentrated at the region of gene recombination. During their maturation, lymphocytes are selected to survive at several checkpoints. Only cells with complete functional antigen receptors are preserved and expanded. In addition, T lymphocytes are positively selected to recognize peptide antigens displayed by self-MHC molecules and to ensure that the recognition of the appropriate type of MHC molecule matches the co-receptor. Immature lymphocytes that strongly recognize self-antigen are negatively selected and prevented from completing their maturation, or in B cell undergo further light chain gene rearrangements that change their specificity, thus eliminating cells with the potential of reacting in harmful ways against self. So the cell mediated immunity activation of T lymphocytes. So there are two T lymphocytes, right, as we have discussed, CD4 positive and CD8 positive T cells. So if we talk about the intracellular microbe, so the phagocytized microbe that survive within the phagolysosome will have two fates, one via CD4 and another via CD8. So if examples include mycobacteria, fungi, protozoans. So if CD4 T cells are nearby or doing their function, CD4 positive T cells that function by secreted cytokines and the co-receptor ligand CD40L, and if CD8 positive T cells that kill the infected cells in which microbe have escaped from the phagolysosome and is present in the cytosol. When we talk about cells or any cell which doesn't have the ability to phagocytize, so and if they are infected, for example, by viruses, by protozoans, then definitely they'll be dealing with CD8 positive T cell, which are the cytotoxic T cells, and they'll directly kill the cell. And these were the induction and effector phases of cell mediated immunity, how basically CD4 and CD8 positive T cells are doing their function. So when naive CD4 positive T cells and CD8 positive T cells recognize peptide that are derived from protein antigens and presented by dendritic cells in peripheral lymphoid organs. The T lymphocytes are stimulated to proliferate and differentiate into effector cells, many of which enter the circulation. Some of the activated CD4 positive T cells remain in the lymph node, migrate into follicles, and help B cells to produce antibodies. Migration of effector T cells and other leukocytes to site of antigen. Effector T cells and other leukocytes migrate through blood vessels in peripheral tissue by binding to endothelial cells and have been activated by cytokines produced in response to infection in these tissues. So here you can connect the concept of inflammation which we read in the beginning, right? So the T cell effector functions, basically CD4 positive T cells recruit and activate phagocytes to destroy microbes, and CD8 positive cytotoxic T cells, they kill the infected cell. So now you can, if we look from the beginning, now you can club the concepts together, right? Earlier they were separate topics, so the help of revision you can actually see where those individual pieces of puzzles are fitting. So this is where you can add the concept of how the cell migrates from blood vessel to the site of infection, and here the site of infection will also be the site of inflammation. And now you can connect the dots of inflammation, what are the things involved in the inflammation, what led to the migration of cells, what were the surface receptors which are involved in it. Okay, so there, those concepts you can connect these things there. Next, so the steps which are involved in the T lymphocytes, basically the maturation of T lymphocytes and how basically they are reaching the site of infection or peripheral tissue, it is listed here. So the naive T cell recognize MHC associated peptide antigens displayed on antigen presenting cells and other cells and other signals. The T cell respond by producing interleukin and expressing receptors for IL-2, leading to an autocrine pathway of cell proliferation. The result in expansion of the clone of T cells that are specific for the antigen. Some of the progeny differentiate into effector cells which serve various functions in cell mediated immunity, and some differentiate into memory cells which survive for long periods. Other changes associated with activation, such as the expression of various surface molecules, can also be associated here, and now we know what such surface molecules will be. They will be cytokine receptors. Okay, and again these are just the type of receptors which we have discussed earlier. Then the antigen recognition and the signal transduction during T cell activation. So now you can understand how the signal transduction is basic. If you have seen this week's lecture, you know what signal transduction, different types of signaling molecules are leading to the change in the biochemical properties of the cell and making them either more susceptible to certain cytokines or being themselves as a ligand for other receptors. Okay, so T cell activation, different T cell molecules recognize antigen and deliver biochemical signals to the interior of the cell as a result of antigen recognition. The CD3 and the epsilon proteins are non-covalently attached, so it's called like zeta, I guess, to the T cell receptor, and alpha and beta chains by interactions between charged amino acids in the transmembrane domain of these proteins. These basically indicates that, okay, if this is a CD4 positive T cell, it will recognize the antigen on the MHC class 2 molecule. This complex will not only have the TCR, they will also have a co-receptor, and then the phosphorylation. So we'll discuss this maybe not today, in the next class, that how the phosphorylation occurs, then what is the signaling thing associated with these changes of the receptor. Then again, these were the few summaries of the signaling pathways which cytokine receptors or TCRs or B cell receptors, they undergo or they initiate such processes in the cell on which they are present. So the overall summary includes: the T lymphocytes are the mediators of the cell mediated arm of the adaptive immune responses, which combats microbes that are ingested by phagocytes and live within these cells, or microbes that infect host cells. T lymphocytes also mediate defense against some extracellular microbes, help B lymphocytes to produce antibodies and destroy cancer cells. The responses of T lymphocytes consist of sequential steps. So, recognition of host cell associated microbes by naive T cells, expansion of the antigen specific clones by proliferation, and differentiation of some of the progeny into effector cells and memory cells. T cells use their antigen receptors to recognize peptide antigens displayed by major histocompatibility complex and APCs, which account for the specificity of the ensuing response and also recognize polymorphic residues of the MHC molecules. Antigen recognition by the TCR triggers signals that are delivered to the interior of the cell by molecules associated with the TCR and by the co-receptors that are CD4 or CD8 depending upon the interaction. The binding of the T cells to APCs is enhanced by adhesion molecules, notably the integrins, whose affinity for their ligands is increased by antigen recognition by the TCR. APCs exposed to microbes or to cytokines produce, as part of the innate immune reaction to microbe, express co-stimulators that bind to receptors on TCR and deliver necessary secondary signals or secondary messengers for the activation of the T cell. The biochemical signals triggered in T cells by antigen recognition and co-stimulation result in the activation of various transcription factors that stimulate the expression of genes encoding cytokines, cytokine receptor, and other molecules involved in T cell responses. The TCR signaling pathway involves protein tyrosine kinases, which phosphorylate proteins that become docking sites for additional kinases called the scaffolding proteins. The signaling pathway include calcineurin NFAT, RAS-MAP kinases, and PI3 kinase mTOR pathway. In response to antigen recognition and co-stimulation, T cells secrete cytokines that induce proliferation of the antigen stimulated T cells and mediate the effector function of the T cells. The T cells proliferate following activation by antigens and co-stimulators, resulting in expansion of the antigen specific clones. The survival and proliferation of activated T cells are driven by the growth factor IL-2. Some of the T cells differentiate into effector cells that are responsible for eradicating infections. Okay. Other activated T cells differentiate into memory cells. So you are all aware of the effector cells, effector T cells, effector B cell, memory B cell, and memory T cell. Naive T cell migrate to peripheral lymphoid organs, mainly lymph nodes, draining sites of microbial entry, whereas many of the effector cells generated in lymphoid organs are able to migrate to any site of infection. The pathways of migration of naive and effector T cells are controlled by adhesion molecules and chemokines. The migration of T cell is independent of antigen, but cells that recognize microbial antigens in tissues are retained at these sites. So these are the characteristics of subsets of CD4 positive helper T cells. So they are Th1, Th2, Th17 and TFH. So the cytokines which are associated with these effector T cells is interferon gamma, IL-4, 5-13, IL-17, 22 and 21, 4 and 13. So the responding cells for these cytokines are macrophages, eosinophils, neutrophils and B cells. So this we'll discuss even within 5-10 minutes or in the next class. The properties of the major cytokines produced by CD4 positive helper T cells. So why we have so many interleukins or so many cytokines? So every cytokine will bind to a particular receptor. That particular receptor will finally initiate a cascade of signaling molecules or signal in the cell which will lead to a specific change in that cell. Okay, so for different changes we need different cytokines and different receptors. Okay, this is the basic concept. So the properties of T cell cytokines: they are produced transiently in response to antigen, usually act on same cell that produces the cytokine. Pleiotropism, that is, each cytokine has multiple biological actions. So imagine if we have to attach every single interleukin to every single function, there will be huge amount of interleukins, right? Huge volume of interleukins will also be there. So to avoid that, there are concepts of redundancy, there is concept of antagonism. So all these things ensure that with the limited amount of things which we have, we can have the optimum function. Okay, and then redundancy, as I mentioned. So multiple cytokines may share the same or similar biological activities, but their source will be different. Okay, then we had a discussion about the cooperation between CD4 and CD8. So from here we can understand that though we are reading things separately, they all play together. Okay, so you know that CD4 positive cell will do different function with the help of cytokines, while CD8 positive T cell will directly kill the microbe without the help of cytokines. Then the migration of naive and effector T cells were discussed. So here, you know, for migration you need certain signals, you need certain cytokines, you need certain receptors, you need certain changes in the cells which are being migrated. So all such things was discussed in this. Then the evasion of cell mediated immunity by microbes. So despite having so many checkpoints, there are still n number of ways through which microbes have evolved to escape our immune checkpoints. Okay, so these are few of the microbes for which things have been studied, and with this study and knowledge we were actually able to cope up with the microbe. We were able to make counter mechanisms or counter drug, or find mechanisms which can counter this effect of the microbe. So microbes include mycobacteria, HSV, CMV, EBV, and then pox viruses. So these are few well studied microbes who are known to escape immune responses or our checkpoints, and there is a constant study goes into it to make sure that these microbes are well studied, so that we can use this education and knowledge to come up with far better drugs or molecules or interventions or whatever you want to say based upon the infection. So the summary includes: cell mediated immunity is the arm of adaptive immunity that eradicates infection by cell associated microbes. This form of host defense uses two types of cells: CD4 positive T cells and CD8 positive T cells. CD4 positive T cells can differentiate into subsets of effector cells that make different cytokines. Effector cells of the Th1 subset recognize the antigen of microbe that have been ingested by the macrophages, right? These T cells secrete interferon gamma and express CD40 ligand, which function cooperatively to associate macrophages. Classically, activated macrophages produce substances including ROS, NO, and lysosomal enzymes that killed ingested microbes. Macrophages also produce cytokines that induce inflammation. So this information is important, like which cytokine does what, because these will help in understanding the way that particular cell is reacting to the infection. Th2 cells stimulate eosinophilic inflammation and trigger the alternative pathway of macrophage activation. And TFH cells induce in parallel trigger IgE production. The balance between activation of Th1 and Th2 cells determines the outcome of many infections with Th1 cells, promoting the Th2 cells, suppressing defense against intracellular microbes. CD8 positive T cells differentiate into CTLs that kill infected cells, mainly by inducing apoptosis of the cells. Many pathogenic microbes have evolved mechanisms to resist cell-mediated immunity. These mechanisms include inhibiting phagolysosome fusion, escaping from the vesicles of phagocytes, inhibiting the assembly of class 1 MHC peptide complexes, producing inhibitory cytokines or decoy cytokine receptors, and inactivating T cells, thus prematurely terminating T cell responses. The central and peripheral tolerance of self antigen. So what exactly is mean by tolerance to self antigen? So this is a very important property. Okay, so the immature lymphocytes is specific for self antigen that encounter these antigen in the central lymphoid organs may be deleted because they are recognizing our self antigen. So the B lymphocytes may change their specificity, that is the receptor or especially the CDRs, right? And some T lymphocytes develop into regulatory T cells. Some self-reactive lymphocytes may complete their maturation and enter peripheral tissue. Peripheral tolerance will come from these cells which just entered the peripheral tissue. So the mature lymphocytes that recognizes self-antigen may be suppressed by Tregs, inactivated or deleted. Otherwise, this will lead to disease conditions such as autoimmunity. Okay. So the cell tolerance in the T cells occurs in the thymus during T cell development. Immature T cells with TCRs that strongly bind self-antigen ensures self-reactive T cell die before entering the circulation. So the medullary thymic epithelial cell expresses many tissue restricted antigens. From this you don't need to remember this 100%. The only thing which you need to remember is that there is a level of tolerance which is there in the T cells, which helps in maintaining the balance between the self and the foreign, or say host or the foreign. Okay, so the phases and types of humoral immune responses. So first, what will happen when the humoral immune response is occurring or taking place? So the activation of B lymphocytes that will lead to proliferation and clonal expansion. Then naive B cells express IgM and IgD as membrane bound receptors. Activation requires antigen binding and additional signaling. Antibody secretion. So at this stage the antibodies will be secreted which have same specificity as the B cell receptor that recognize the antigen. Once activated, B cells can produce thousands of plasma cells, each secreting thousands of antibodies per hour. Then the class switching will take place. So some B cells change antibody heavy chain class, that is IgG, IgA and IgE, enabling different effector functions against different microbes. Affinity maturation will then take place. So during the immune response, antibodies improve their binding strength to antigen, enhancing their ability to neutralize microbes and toxins. Antibody responses are T dependent or T independent. Okay, so the T dependent responses are triggered by protein antigens. They require helper T cells which recognize the same protein, lead to class switching, affinity maturation, and then the memory B cells produce strong, specialized and long lasting antibody responses. In T independent responses, they are triggered by non-protein antigens. So it can be bacterial surface molecules, which can be polysaccharides, lipids, nucleic acids, multivalent antigens and other things as well. They do not need T cells. So B cells are directly activated with innate signals. They are responsible for short-lived. They are not that specific, so weaker in nature, and they don't lead to the further maturation of the receptor. So the class switching or the affinity maturation is lacking in T independent maturation. Follicular B cells, so they basically reside in the lymphoid follicles and recirculate, mediate T dependent responses and produce class switched high affinity antibodies. So we'll see the figure in the coming slide. Then the marginal zones, what are these? So they are found in the splenic marginal zone and lymph nodes outer follicle rims. So this is the arrangement of cells which are present in the lymphoid tissue. Respond mainly to blood-borne polysaccharides and lipid antigens. produce T independent IgM antibodies with limited receptor diversity. B1 cells, so these are located in mucosal tissues and peritoneum, respond mostly to multivalent polysaccharide and lipid antigens. Also produce T independent IgM antibodies. This is the same thing, like this is basically the summary of the response. So we can see the same thing which are written here in the table. So the exposure, so in the primary response, this is basically happens when the system sees the antigen for the very first time. Secondary response is the subsequent encounter. The lag phase generally lasts for 7 to 10 days in primary response, while it lasts only for 1 to 3 days thanks to the memory cells in the secondary response. Antibody quantity is very low in primary response and it is of IgM type, while in secondary response the antibody quantity is high and is majorly IgG, IgA or IgE depending upon the type of infection. Then affinity is low in primary response and high in secondary response. Duration for the primary response is short-lived and for secondary response it is long-lived. The similar things are depicted here in a different way, how basically the cells are being matured in the bone marrow. Naive cells come to the secondary lymphoid organ and then they come to the peripheral lymphoid organs. Okay, then this is the T dependent and T independent antibody responses. So in T dependent, you can clearly see that a T helper cell will be associated with the B cell which has just recognized the antigen, and then the class switching, affinity maturation, formation of memory B cells will take place. While in T independent one, the B cell will directly recognize the antigen, which need not necessarily be a protein entity. Okay, it can be any polysaccharide molecule, lectin or anything. Then it will generate other signals which will lead to the formation of secretory IgM, which are low affinity and they are short lived in the system. Again, as I have mentioned earlier, beginning of the class, that this is an important graph to know how the primary and secondary response take place, what sort of antibodies are present in these phases, and how long exactly these phases last. So if we talk about the primary response, the lag phase is usually for 5 to 10 days, while for secondary response it is for 1 to 3 days. The peak response or the maximum response is smaller when we compare to the response which is there in the secondary response. There is generally IgM is present more in comparison to IgG in primary response, while in secondary response IgG is IgG, IgA or IgE. So take any antibody again with respect to the antigen will be more in comparison to the IgM type of antibody. Okay, so the affinity of the antibody in the primary response is low and is not that specific when we are comparing it to secondary response, but this will still be more specific when we are comparing it with the innate immunity. Okay, innate immunity is completely non-specific. While we compare primary antibody response to secondary antibody response, in comparison to secondary antibody response, primary antibodies responses affinity is low. It is still high in comparison to the whole complete affinity with respect to the innate immunity. Okay, then antigen receptor mediated signal transduction in B lymphocytes was also discussed. Again, these are the signaling molecules, these one, and these are the transcription factors. So if you have taken the current week classes and you have completed it, you know what these entities or what these molecules are doing and how they are doing and how it is affecting the cell's behavior overall. Okay, then the mechanism of helper T cell mediated activation of B cell, again it is the same thing. Why we are repeating it multiple times? So that it can be very well understood and we have a whole picture of everything. Okay, Then again, the germinal center, it is one of the another important concepts of immunology. So the germinal center reaction, like what happens in the germinal center. So the B cells that have been activated by T helper cell at the edge of a primary follicle migrate into the follicle and proliferate, forming the dark zone of the germinal center. Germinal center B cell in the dark zone mutate their IgE genes, and at extremely high rate, a process called somatic hypermutation, and migrate into the light zone, where B cells with the highest affinity Ig receptors are selected to survive, and they differentiate into plasma cells or memory cells. So you should remember what is present in the dark zone, what is present in the light zone, and what exactly exit the system. Okay. Then the immunoglobulin heavy chain class switching or the isotype switching. So if you have different signaling molecules to different B cells, you will get different type of antibody. Okay. So antigen stimulated B lymphocytes may differentiate into IgM antibody secreting cells, or under the influence of CD40 ligand and cytokines, some of the B cell may differentiate into cells that produce different Ig heavy chain classes, that is isotype switching. The principal effector function of some of these isotypes are listed in the table above. All isotypes may function to neutralize microbe and toxins. B cell activating factor belonging to the TNF family, example BAF, is a cytokine that may be involved in switching to IgA. So if the activated B cell got BAF signal, then it will secrete IgA. Okay. Especially in T dependent response. Switching to IgG subclass is stimulated by the cytokine interferon gamma in mice, but in humans it is thought to be stimulated by other cytokines. So as I've mentioned earlier as well, that these are few things which are still, like, whatever we know, it is still very less what actually happens. Okay, so it is a active area of research which goes on. Then these are again the similar concept of germinal center was discussed, and again how this was just the revision for the things between, like, what happens at every stage of the maturation of the B cell. And this is same depicting the similar thing in a different way, that when the antigen binds, then the downstream signaling and like what are the major pathways, how it changes. So these things were discussed. So I'll just go through the summary very quickly. So the humoral immunity is mediated by antibodies that bind to extracellular microbes and their toxins, which are neutralized or targeted for destruction by phagocytes and the complement system. Humoral immune responses are initiated by recognition of the antigens by specific membrane immunoglobulin antigen receptors of the naive B cell. The binding of multivalent antigen cross-linked B cell antigen receptors of specific B cells, and biochemical signals are delivered to the inside of the B cells by Ig associated signaling proteins. In humoral immune responses to a protein antigen called T-dependent responses, the binding of protein antigen to a specific Ig receptor of naive B cell in lymphoid follicle results in the generation of signals that prepare the B cell for interaction with activated helper T cell that expresses CD40L and secretes cytokines. The B cell internalize the process that antigen and present class 2 major histocompatibility complex. These helper T cells contribute to early B cell activation at extrafollicular sites. The early T-dependent humoral response occurs in extrafollicular foci and generate low levels of class-switched antibodies. Activated B cell induce the further activation of T cells and their differentiation into T follicular helper cells. The B cell together with the TFH cells migrate into follicles and form germinal centers. Affinity maturation is the process by which the affinity of antibodies for protein antigen increases with prolonged or repeated exposure to the antigens. The process is initiated by signals from TFH cells, resulting in migration of the B cell into follicles and the formation of germinal centers. Here the cells proliferate rapidly and their IgV genes undergo extensive somatic mutation. The antigen may be displayed by FDCs in the germinal centers. B cells with mutated V regions that recognize the antigen with high affinity are selected to survive, giving rise to affinity maturation of the antibody response. Polysaccharides, lipids, and other non-protein antigens are called T-independent antigens because they induce antibody responses without T cell help. Most T-independent antigen contain multiple identical epitopes that are able to cross-link many Ig receptors on a B cell, providing signals that stimulate B cell responses even in the absence of helper T cell activation. Secreted antibodies form immune complexes with residual antigen and shut off B cell activation by engaging in the Fc receptor of the B cell. The same thing which we have just read, it is depicted here in a little bit more concise way. Okay, then okay, so I guess, yeah, we have almost covered things. So these are just similar things, like what is the role of antibodies. We have seen them in the last class itself, and then we started with the complement system. So in the next class we're gonna start with the complement system, but for now we're gonna see some of the helping questions. So if I'm correct, I guess we have covered this. I remember this. We have covered this as well. Yeah, so okay, we're gonna start from here. So the first question for today, and obviously so the questions are pretty simple. So though I say 1 minute, but it's just 30 seconds, so just put your answers in the chat box. Okay, so the question says pro B cells mature into pre B cells upon interaction of VLA4 and VCAM1, CKIT and SCF, both A and B, or FLT3 and FLT3L. Your time starts now. Just put your answers in the chat box. The right answer is C, both A and B. So just for your revision purposes, we're gonna see this because it's been a while since we have discussed this, right?
Hi everyone, in this video I would discuss the B cell development. Now, B cell development starts in the bone marrow. Bone marrow is the primary lymphoid organ where immune cells develop. In the early stage of B cell development, it takes place in the bone marrow. Then later, when B cells mature a little bit, it moves out to the secondary lymphoid organs such as lymph node and develops there. So the primary place of B cell development is the bone marrow. Inside the bone marrow, there is endosteal niche. Endosteal niche is closer to the bones, so where the osteocyte.
I feel we have discussed this as well. Okay. So the next question. So heavy chain rearrangement occurs in hematopoietic stem cell, multipotent progenitor cell, pro-B cell or pre-B cell. We're gonna go a little bit quickly for these questions because we have questions from complement part as well. So like quickly give your answers in the chat box and we can move forward. So remember these, so from these questions as well you can get an idea on what are the important things which you should keep in your mind while reading a topic, right? So for example, I mentioned about the cytokines and their responses. Here the site of affinity maturation, somatic hypermutation, class switching, where exactly they are happening and what is happening in them. So these events are important to remember. Okay. So the right answer, that heavy chain rearrangement occurs in the pro-B cells. So here is the basic development of it. I'm gonna complete this part because this is important to remember. So take this as a revision again.
Dave explains. As we learned in the previous tutorial, the adaptive immune system utilizes two major types of immune cells, or lymphocytes, those being T cells and B cells. These cells can react to a tremendous variety of antigens, for which these cells possess a large variety of cell surface receptors that can recognize an unlimited number of pathogens. To be able to generate such an astounding repertoire of receptors, BCR and TCR, or B and T cell receptor genes, are created by recombining pre-existing gene segments. VDJ recombination, or recombination of variable V, diversity D, and joining J gene segments, is an essential step in the development of the vertebrate immune system. DNA rearrangements that generate functional immunoglobulin, IG, and T cell receptor proteins are indispensable for the development, progression, and even the survival of B and T cell precursors. This is initiated by breakage at precisely defined locations in DNA, and the joining is completed by the common cellular repair response, related to the repair of breakage caused by factors such as ionizing radiation or other genotoxic agents. These VDJ gene fragments are typically found as multiple copies that lie transcriptionally upstream of a constant region, C. During the development of the lymphoid cells, segments of each type are joined by VDJ recombination so as to make a VJ or VDJ array that codes for a variable region exon, which becomes linked by RNA splicing to the C region. Rearrangements occur in an ordered fashion, with D to J joining preceding before a V segment is joined to the rearranged DJ segment. Immunoglobulins, or IGs, are produced by B lymphocytes and secreted into plasma. The IG molecule in monomeric form is a glycoprotein that is shaped more or less like a Y. Basic structure of the IG monomer consists of two identical halves connected by two disulfide bonds. Each half is made up of a heavy chain and a light chain. The IG heavy chains are encoded by recombined VDJ genes, while VJ rearrangements encode the immunoglobulin light chains. T cell receptors, or TCRs, are membrane-bound heterodimeric proteins normally consisting of the highly variable alpha and beta chains. T cells expressing these two chains are referred to as alpha-beta T cells, though a minority of T cells express an alternate receptor formed by variable gamma and delta chains, referred to as gamma-delta T cells. TCR beta chains and delta chains are similarly encoded by distinct sets of V, D, and J genes, while alpha chains and gamma chains are encoded by additional sets of V and J genes. The number of possible IG or TCR molecules has been estimated to be well above 10 million. This feat is accomplished by a highly regulated and well-coordinated set of reactions that begins with cleaving DNA within specific well-conserved recombination signal sequences, RSSs, that are adjacent to each V, D, and J coding segment. RSSs consist of conserved heptamer and nonamer elements, separated by 12 or 23 nucleotides of less conserved spacer sequences, and their relative orientation determines whether the reaction proceeds by inversion or by deletion. In vivo, recombination takes place between one RSS with a 12 base pair spacer and one with a 23 base pair spacer. This is the so-called 12-23 rule. The cleavage phase begins with RAG1 and RAG2, enzymes which work as a multi-subunit complex to induce cleavage of double-stranded DNA. These enzymes bind to either a 12 RSS or 23 RSS. This complex then synapses with a partner RSS of the complementary spacer length. The RAG complex then nicks one DNA strand of each RSS and catalyzes transesterification reactions to generate hairpinned coding ends and blunt signal ends. In the joining phase, both the coding and signal ends are repaired by non-homologous end joining, or NHEJ, the main pathway for repair of DNA double-strand breaks. But here, the coding ends are often repaired imprecisely to increase diversity, as indicated by the red nucleotides in between here, while the coding ends undergo extensive processing, resulting in addition or deletion of bases to increase antigen receptor gene diversity. The RSSs at the signal ends are precisely joined in a head-to-head arrangement, generating a signal joint, or SJ. Usually, VDJ recombination deletes the DNA between the gene segments to generate an excised signal circle, or ECS, that is covalently sealed at the SJ. The ECS is lost during cell division. Unfortunately, errors can arise during this process. They can be broadly classified into joining and recognition errors that can result in generating deleterious genomic rearrangements. Mice with disruptions of either the RAG1 or RAG2 gene are completely defective in VDJ recombination and therefore possess no mature B or T cells. These mice have no other obvious defects, which implies that the RAG genes function only in the immune system. With T and B cell development better understood, let's move forward and learn about their activation next. Thanks for watching. Subscribe to my channel.
So the next question for today. Match the B cell stage with the proper description. Early pro B cell, smaller pre B cell, immature B cell, late pro B cell, larger pre B cell. VDJ rearranging, DJ rearranging, expressing pre B cell, VJ rearranging, and surface IgM. So it's a little bit big question. So I'm gonna give you guys 1 minute for this, and your time starts now. Try to remember the process and the stages of those cells. So the right answer is A, that is early pro B cell will have DJ rearrangement, small pre B cell will have VJ rearrangement, immature B cells will have surface IgM, late pro B cells will have VDJ rearranging, and then finally large pre B cell will have VJ rearrangement. B cell activation occurs in, this is a pretty easy question, spleen, lymph node, thymus, bone marrow. So the right answer is lymph node. And as this is an important topic, we're gonna take the help of visual aid so that our concepts are more clear and we remember them.
Science Stop! Surface receptors that can recognize an unlimited number of pathogens. To be able to generate such an astounding repertoire of receptors, BCR and TCR, or B and T cell receptor genes, are created by recombining pre-existing gene segments. VDJ recombination, or recombination of variable V, diversity D, and joining J gene segments, is an essential step in the development of the vertebrate immune system. DNA rearrangements that generate functional immunoglobulin, Ig, and T cell receptor proteins are indispensable for the development, progression, and even the survival of B and T cell precursors. This is initiated by breakage at precisely defined locations in DNA, and the joining is completed by the common cellular repair response, related to the repair of breakage caused by factors such as ionizing radiation or other genotoxic agents. These VDJ gene fragments are typically found as multiple copies that lie transcriptionally upstream of a constant region, C. During the development of the lymphoid cells, segments of each type are joined by VDJ recombination so as to make a VJ or VDJ array that codes for a variable region exon, which becomes linked by RNA splicing to the C region. Rearrangements occur in an ordered fashion, with D to J joining preceding before a V segment is joined to the rearranged DJ segments. Immunoglobulins, or IGs, are produced by B lymphocytes and secreted into plasma. The Ig molecule in monomeric form is a glycoprotein that is shaped more or less like a Y. Basic structure of the Ig monomer consists of two identical halves connected by two disulfide bonds. Each half is made up of a heavy chain and a light chain. The Ig heavy chains are encoded by recombined VDJ genes, while VJ rearrangements encode the immunoglobulin light chains. T cell receptors, or TCRs, are membrane-bound heterodimeric proteins normally consisting of the highly variable alpha and beta chains. T cells expressing these two chains are referred to as alpha-beta T cells, though a minority of T cells express an alternate receptor formed by variable gamma and delta chains, referred to as gamma-delta T cells. TCR beta chains and delta chains are similarly encoded by distinct sets of V, D, and J genes, while alpha chains and gamma chains are encoded by additional sets of V and J genes. The number of possible Ig or TCR molecules has been estimated to be well above 10 million. This feat is accomplished by a highly regulated and well-coordinated set of reactions that begins with cleaving DNA within specific well-conserved recombination signal sequences, RSSs, that are adjacent to each V, D, and J coding segment. RSSs consist of conserved heptamer and nonamer elements, separated by 12 or 23 nucleotides of less conserved spacer sequences, and their relative orientation determines whether the reaction proceeds by inversion or by deletion. In vivo, recombination takes place between one RSS with a 12 base pair spacer and one with a 23 base pair spacer. This is the so-called 12-23 rule. The cleavage phase begins with RAG1 and RAG2, enzymes which work as a multi-subunit complex to induce cleavage of double-stranded DNA. These enzymes bind to either a 12 RSS or 23 RSS. This complex then synapses with a partner RSS of the complementary spacer length. The RAG complex then nicks one DNA strand of each RSS and catalyzes transesterification reactions to generate hairpinned coding ends and blunt signal ends. In the joining phase, both the coding and signal ends are repaired by non-homologous end joining, or NHEJ, the main pathway for repair of DNA double-strand breaks. But here, the coding ends are often repaired imprecisely to increase diversity, as indicated by the red nucleotides in between here, while the coding ends undergo extensive processing, resulting in addition or deletion of bases to increase antigen receptor gene diversity. The RSSs at the signal ends are precisely joined in a head-to-head arrangement, generating a signal joint, or SJ. Usually, VDJ recombination deletes the DNA between the gene segments to generate an excised signal circle, or ECS, that is covalently sealed at the SJ. The ECS is lost during cell division. Unfortunately, errors can arise during this process. They can be broadly classified into joining and recognition errors that can result in generating deleterious genomic rearrangements. Mice with disruptions of either the RAG1 or RAG2 gene are completely defective in VDJ recombination and therefore possess no mature B or T cells. These mice have no other obvious defects, which implies that the RAG genes function only in the immune system. With T and B cell development better understood, let's move forward and learn about their activation next.
Thanks for watching. Subscribe to my channel.
Moving to the next question. So the protein antigens can induce B cell activation by thymus independent process, thymus dependent process, both A and B, or T helper cells. So we just covered it. It's a pretty simple question. So I'm gonna give you guys 30 seconds for this question. Please write your answers in the chat box. Answer is thymus dependent process, right? So this doesn't need any explanation. Next question for today is, given are the steps for maturation of progenitor B cells into precursor B cells, the correct sequence of events are binding of c-Kit on the pro-B cell to stem cell factor, binding of IL-7, binding of VCAM1, expression of IL-7, and then maturation of pro-B cell. Please go through this question carefully. Remember the whole process of B cell development and then try to answer this. So I'm going to give you guys 1 minute or 1 minute 30 seconds for this particular question because it's a little bit descriptive in nature. Okay, and your time starts now. correct sequence is B, that is binding of VCAM1 on the stromal cells in VLA4 on the pro-B cell, binding of c-KIT on the pro-B cell to stem cell factor, expression of IL-7 receptors on the pro-B cell, binding of IL-7 released from the stromal cell to the IL-7 receptor, and maturation of pro-B cell to pre-B cells. Next question for today. Examples of type 2 thymus independent antigen are, so remember it is asking for thymus independent. Will it be polysaccharide, bacterial flagellin, bacterial cell wall polysaccharides, both A, B, or both B and C? So the correct answer is E, that is both B and C. Remember, except the protein, it will recognize the bacterial surface things. So if you can look at the event, I have pasted here the same thing which we have read, is just this is little bit in different way, and how it is basically recognizing those things are present there. So the somatic hypermutation of heavy and light chain variable region genes occur in light zone, dark zone and light zone respectively, light zone or dark zone. So it's a pretty straightforward questions. Try to put your answer in the chat box, and I guess I'll give 30 seconds for this. It's pretty easy, right? The right answer for this particular question is, like, it's pretty straightforward, right? So it's the D. So if you remember, this is the representation of light zone and dark zone, same thing, different way of representation. Okay, so one question, like few more questions to go. Okay, so what is the initial complement component bound by complement fixing antibodies? Is it C1s, C3b, C1q, or none of these? So 30 seconds for this particular question. the right answer is C, C1Q. So this is the structure of C1Q. If you have completed the complement system, you might be knowing like which pathway exactly we are talking about. Okay, so I guess we'll start with the complement system in the next class a little bit in more details, and then we'll take the questions from there onwards. Okay, so thank you so much for your patience. See you all in the next class. Bye bye.